Okay, this is part B of chapter 27. The penis has a long shaft or body containing 3 columns of erectile tissue. The 2 larger ones are called the corpora cavernosa. They're on the dorsal side of the penis. The smaller ventral side column is called the corpus spongiosum. So individually, you have a corpus cavernosum. And yes, this is the dorsal side of the penis. This is the ventral side of the penis. So the penis is like your feet. When you look down at the top of your foot, that's actually the dorsal side because it has the nails like your hands in the correct anatomical positions, palms are forward. So the penis and the feet are alike when we look at ventral and dorsal, or posterior and inferior. The expanded distal end of the penis is called the glans penis. It has numerous sensory receptors, which is why it's typically covered by the prepuce, which is modified skin to prevent constant stimulation. However, this is often removed in a circumcision. The circumcision is going to remove the tissue. There are religious reasons, there are hygiene reasons, cultural reasons for the circumcision. Now, don't wait until you're in the middle of labor and think you're going to get what you want for your child. You have to make those decisions before you go into labor and prepare ahead of time. If you don't know you're having a boy, you still need to make those preparations just in case you do have a boy. Anyway, there are different techniques. There are different doctors that do this. Sometimes it's a pediatrician. Sometimes it's the OBGYN or obstetrician. The procedure could involve a scalpel being used to remove the prepuce, or it could be a two-part ring that's put in place. It's plastic and it cuts off the circulation and the prepuce falls off later. With a scalpel, sometimes they use anesthesia and sometimes they don't. So you need to, again, check that out, and that way your wishes can be followed. The circumcision can be done at any time. There are some cultures where it's removed as a rite of passage into adulthood. If it's not removed, there has to be additional hygiene because the prepuce can become a reservoir for bacteria, fungus, and debris. So with the prepuce in place, it would have to be rolled back, the glans cleansed and dried, and then put the prepuce back in place. So we have meiosis. Meiosis is a way of dividing up the nucleus. Mitosis is the other way that the nucleus can be divided. So meiosis is defined as a type of nuclear division that produces gametes. They're 9 identical cells with only half of the genetic material needed for a new individual. Gametes are what the cells that are required for sexual reproduction. Terms associated with DNA. Chromatids are single strands of DNA or single molecules of DNA. They use the term chrome because they can easily pick up the dye since they're half DNA and half proteins. So they're easily visible in a stained cell. Chromatin or chromatin refers to the long stringy version of DNA, which is present when the cell's not dividing. And that's why the nucleus is typically the darkest stained organelle in the cell. Chromosomes is the tightly coiled version of DNA. And that's what we see, here and here. If I move this thing down. So, chromosomes, chromatin, molecules of DNA. Diploid refers to the amount of DNA found in typical cells. It's called the full amount. It's represented as 2N. In humans, 2N would be 46 or 23 pairs. If we were just talking about Drosophila or fruit flies, that would be 12 pairs. Or rather, 12 chromosomes, which is 6 pairs. And so fruit flies and human beings can't reproduce because they don't have the same number of DNA molecules. Haploid refers to the number of chromosomes found in the gametes, which would be the sperm and the ova. Ova is plural for ovum. This would be half of the diploid amount. So it's N. So in humans, N would be the 23. Then you have some blanks to fill in. Chromosomes have one chromatid, whereas replicated chromosomes have 2 chromatids. And so we also refer to them as single-stranded and double-stranded chromosomes. Spermatogenesis and spermiogenesis. We need to separate the two. Spermatogenesis is the production of sperm, and that's all of this. Happens within the testes, regulated by the follicle stimulating hormone that comes out of the anterior pituitary gland. Spermiogenesis is the last part of spermatogenesis. It's the maturation of the spermatids, which are these cells, into the spermatozoa or sperm. This takes place in the testes, but also finishes up in the epididymis. Okay, we're going to name and describe the cells produced along the process of spermatogenesis. The spermatogonium is the first cell, and you have a table to fill in. There's one of those. It has the diploid amount of DNA with one strand or single-stranded. That's going to be a cell that because of follicle stimulating hormone, is going to mature into a primary spermatocyte. The primary spermatocyte, there's also going to be one of those. It has a diploid amount of DNA, but it has double-stranded or two strands of DNA. That cell, again, because of follicle stimulating hormone, is going to undergo meiosis 1. And we're going to generate 2 secondary spermatocytes. It has the haploid amount of DNA. So that's the difference between a primary and a secondary spermatocyte, is the volume of DNA. The strands are still going to be 2 because you've still got them together. Each secondary spermatocyte undergoes meiosis 2, so that's going to generate a total of 4 spermatids. Spermatids are also haploid, half the genetic material, but they're going to have one strand or be single-stranded. They're a generic looking cell, just like the rest of them. Spermiogenesis, so because of testosterone, each individual spermatid is going to become a spermatozoan, which we usually refer to as sperm. It has the same genetic makeup, haploid amount, with one chromosome. So we start with one cell and we wind up with 4. Okay. The product is a spermatogonium, or spermatozoan, sorry. The spermatozoan is a cell that has a head, which has the nucleus with the 23 chromosomes. That's the only part of the sperm that is going to become part of the zygote. Okay, so covering the head of the sperm is the acrosome, and that's an enzyme layer needed to penetrate the corona radiata. Oops. Corona radiata, which is going to be surrounding radiata. Yeah. Sorry, radiata. That's going to be the layer of tissue surrounding the developing ova. The acrosome is going to make contact with the corona radiata and prevent other sperm from penetrating. So when the corona radiata is contacted with the acrosome, sets off a trigger of chemical reactions. Okay, so attached to the head is the body or mid piece, and that's where all the mitochondria are going to be produced. And that are the mitochondria are found. So the mitochondria are going to convert fructose into ATP so that you've got energy for the sperm to propel the tail. The tail is the flagellum, which is enacted or stimulated by the prostaglandins in sperm to propel the sperm through the female reproductive system. Okay, components of semen. Fructose provides energy for sperm motility. Seminogelin thickens the semen so it adheres to the vaginal wall. Prostaglandins stimulate the motility of sperm and they cause female peristalsis. So, to shorten the distance that the sperm has to travel, you have female peristaltic contractions to move the developing ovum closer to the uterus. Serine protease is going to liquefy the semen within 30 minutes. And that's about how long it takes for the prostaglandins to trigger the motility of the sperm. So like I said, the sperm can move the flagellum prior to being exposed to semen, but it can't swim. So by the time it's able to swim, now it's got a liquid environment to swim through. Spermine is a term for the collective alkaline or base components to deal with the acidic environment in the vaginal canal. Normal sperm count is somewhere between 50 million and 120 million per microliter. So less than 50 million sperm per microliter is considered a low sperm count. So the E terms. Erection is a parasympathetic response to visual and or tactile stimulation. Columns of erectile tissue are going to fill with blood. Because it's a parasympathetic response, it's not a response to stress. It can happen when you're awake. It can happen when you're asleep. The visual or tactile stimulation can be actual, real, or they could be implied or imagined. Sexual arousal sometimes associated with this, but not necessarily. It could be a response to something else. Emission is a sympathetic response to cause the release of seminal vesicle, prostate, and vulval urethral fluids, as well as to move sperm into the urethra. So this would be going beyond erection or after an erection has occurred. Tactile, visual stimuli, increasing the sensory receptors. Ejaculation is also a sympathetic response, and this is going to push the semen out of the body. So, emission and ejaculation are considered stress responses, but erection is not a stress response. Okay, that's the end of chapter 27 on the male reproductive system.